Welcome, everyone. So welcome to today's virtual seminar. Today's speaker is Dr. Jakob von Moltke. And it's my great pleasure to introduce Jakob. Jakob received his bachelor from Dartmouth College and his PhD from UC Berkeley. So after obtaining his PhD, he moved across the town to UCSF and worked with Dr. Richard Leukotrienes for his postdoctoral training, where he made this landmark discovery that Tuft cells trigger type 2 immunity in response to a worm infection. In 2017, Jakob joined University of Washington as a faculty member. Since then, his lab has been very productive and published several papers describing how Tuft cells detect and respond to worm infection. He's not only like prolific, but he is also very good at attracting funds to support his research. He's currently supported by a DP2 grant as well as a new R1 grant. He also has several foundation grants. So what actually impressed me most is his openness. So Jakob is always willing to share his ideas, data with us because we cross paths. We are also interested in the how Tuft cell work because Tuft cell express a lot of taste signaling elements. So without further ado, I'll let Jakob take over. Jakob, welcome to Monell's virtual seminar.
Great, thank you, Peiwa. I'll start sharing my screen here. Thanks for the invitation. I'm really delighted to be able to give this presentation. You know, you'll see my background is really in immunology, and we stumbled into studying chemosensation and Tuft cells. But I really kind of view the Monell Senses Center as a kind of a mecca for a lot of that work and for the foundational work that was done on Tuft cells originally. So it's great for me. I'm sorry I couldn't sort of make the pilgrimage in person, but it's great to interact with all of you, at least this way. So in these changed times, I've made a little extra space here where you can put the video if you want to keep that, and I'll try to leave that open on all my slides. So I'm going to talk about Tuft cells in the intestine today, but let me just start out by saying that very generally, and a question that has interested me for a long time is how the so-called type 2 immune response to parasitic worms and allergens is initiated. So what it is that happens in the very first moments when the immune system encounters these kinds of agonists. And related to that, the question of how and why an enormous diversity of agonists all filter down into this same type 2 immune response. So, you know, what we're learning is that there's, of course, no single answer, no single pathway that explains this. There's not even a single paradigm. In particular, it really seems to depend a lot on the tissue in which you're asking this question. And I'm going to talk just exclusively about the small intestine today. So very quickly, let me, by way of introduction, just remind you that you have the large villi here that form the absorptive capacity of the intestine, and down below you have the more numerous but smaller crypts. And it's down at the base of the crypts here that the stem cells reside. They're intercalated with Paneth cells and give rise to undifferentiated epithelium that expands further in the transit amplifying zone. And these cells are constantly moving upwards towards the tips of the villi, where they will then slough off. And as they move up, especially as they emerge out of the crypts, they adopt their terminal fates. And Most of these cells will become absorptive enterocytes, but there are also these less frequent lineages, sometimes called the secretory lineages, although that's not quite the right term. That includes the Paneth cells, the goblet cells that make mucus, enteroendocrine cells, and Tuft cells as well. And I guess the only other thing I'll mention here is that the turnover here is incredibly rapid. Half-life is about 3 or 4 days, both in mice and humans. So this entire epithelium is being replaced constantly, and therefore also offers opportunity for very rapid remodeling and change. The other thing I want to introduce before I sort of go further is this worm model that we rely a lot upon. It's called Nipostrongylus brasiliensis. It's an excellent model of human hookworm, has the same life cycle in rodents. The infection is with larvae through the skin, and then it traffics into the lungs where it will molt once, and then crawl through the tissue and eventually be actually coughed up out of the trachea, swallowed back down the esophagus, and find its way to the proximal small intestine, the duodenum predominantly, where it finishes its maturation. And these worms then mate and start to lay eggs that pass out into the environment. So that's a rat-adapted parasite actually, and induces a very nice type 2 immune response that is cleared from mice. Okay, so again, thinking about this question of initiation of these immune responses, this is usually happening at a barrier site with an epithelium. And if we kind of think about the end result, we've known for a long time that the output is what's called weep and sweep, this expansion of the number of mucus secreting goblet cells, changes in the smooth muscle function so that the intestine actually becomes more contractile and therefore is better at expelling the worms. And all of this is driven predominantly in the tissue by the cytokine IL-13. And we've known for a long time that type 2 helpers, Th2 helper cells can make IL-13, but these are adaptive cells and they don't live in the tissue, and they therefore need to be activated in lymph nodes, recruited to the tissue. All this takes time. So in the context of initiation, the discovery of these group 2 innate lymphoid cells, or ILC2s, by the Loxley lab and others was really important because these cells are placed into the tissue during development, and they reside there for your entire life. And they therefore can be some of the first cells that respond in the initiation of type 2 immunity. And so the question really is, how do you get from here, from these agonists, to activating these immune cells in the underlying tissue? And really the approach has been to think about this by kind of working backwards from the ILC2s and asking what's happening upstream. Okay, so this is an overwhelming depiction of all of the receptors that have now been identified to contribute to the activation of an ILC2. I'll come back to say a little bit more about this later, but right now I just want to say that what started all of this is that we wanted to understand better this IL-25 receptor and the cytokine IL-25 that is its ligand. And we knew from previous work that if you take a wild-type mouse and infect it with this Nipostrongylus worm, it will clear that worm by 10 days. But an IL-25 knockout mouse was shown to have a big defect in clearance, so it's still fully colonized 10 days post-infection. The thing that we didn't understand was really where was the IL-25 coming from and how was it being regulated? And that's generally true for a lot of these signals that activate the ILC2s. And so the way that we went about trying to address that is by making a reporter mouse, which the Loxley lab really helped to pioneer as a technique. In this case, we put an IRS RFP downstream of the IL-25 coding sequence without disrupting its function. And then we went looking for red cells and found them in actually lots of places in the body, barrier tissues mostly. Again, I'm just focusing on the intestine, where you can see as an example here, the duodenum. Green is epcam, so it marks the epithelium. Blue is the nuclei, and then there are these rare red IL-25 producing cells. And so we, of course, immediately wanted to understand what those are, and in data that I'm not going to show, what we found is that they are all Tuft cells. And in fact, it seems that every IL-25 producing cell is a Tuft cell, and every Tuft cell makes IL-25. All right, so at that point, we had to go scramble to the literature and find out what Tuft cells are because unlike some of you, we had never heard of them. So But we've just realized then that they were discovered long ago, about 60 years ago, when people started taking electron microscope pictures like this one and noticing these cells in the epithelial barriers with this unique morphology defined really by this so-called tuft, these long, blunt microvilli at the apical surface here. Other interesting features as well, but this is what stands out immediately. And in the scanning EM that we did with some collaborators at Salk recently, you can just see how these tuft cells really are distinctly raised above their neighboring epithelium, which is a really interesting feature, although its function is at this point, as far as I know, remains unknown. Okay, so now you can, after a lot of early EM studies that really defined the morphology of the cells, you can draw this kind of cartoon depiction with the tuft at the top, some intriguing extensions, lateral extensions into neighboring cells, actin bundles, microtubule, and vesicles. The cell biology really is largely still a black box. What we do know is that they're generally rare cells found in the epithelium of hollow endoderm-derived organs, and that includes the intestines, the upper airways, the urethra. I will say right now, and this is thanks in large part to Ichiro Matsumoto's work at Monell, we know that POU2F3 is a master transcription factor for tuft cells, and that'll be important later because the knockouts, I think you maybe think of these as skin 1A, but the knockout has no tuft cells. But then for decades, really, the function of these cells remained mysterious. And that people started to unlock that maybe 10, 20 years ago. A lot of people I see, names I see here on this seminar who really started, especially in the airways, people like Noam Cohen, Bob Margolski, Marco Tizzano, who really started to realize that these have important functions in the airways, in particular sensing bacteria and maybe regulating evasive behavior. But in the intestines, there was very little known about their function. And so we were coming at this from an immunology perspective and a worm perspective. And so we wanted to ask, well, what happens during worm infection? And the really remarkable discovery was that if you compare an uninfected small intestine here staining for tuft cells with this marker DCLK1, you see 1, 2, 3, just very rare cells in 2 villi. But 7 days post-infection, you have this incredible explosion of tuft cells, a hyperplasia with dozens in a single villus. And we can quantify this by taking pictures and counting the number of tuft cells per millimeter of crypt villus. And it just gives us this really nice readout for this type 2 immune response in the epithelium. And I'm just showing that here so that you'll see this over and over again as a quantification of the immune response by counting the tuft cells. Okay, so at the time then what we published was this, what we call the Tuft ILC2 circuit. And the way that works is that during worm infection, tuft cells contribute to this response by making IL-25. That IL-25 activates the ILC2s, and they start to make these canonical type 2 immune cytokines like IL-5, IL-9, IL-13 that drive all of the kind of downstream hallmarks of type 2 immunity: eosinophilia, activation of mast cells, the mucus overproduction that I mentioned earlier. But one of the important things that I want to highlight is that IL-13, among its many targets, its receptors are expressed on the undifferentiated epithelial cells down here in the crypt. And we don't understand how this works, but IL-13 signaling in these cells somehow biases their lineage commitment towards tuft cells, so that in the presence of IL-13, this stem cell compartment starts to produce way more tuft cells, and that's what gives rise to the hyperplasia that I just showed you. And so you get a remodeling within a few days where you completely change the composition of your intestinal epithelium towards this. So this was work that we did, and at the same time as others. Sort of what I really like about it is that all of us arrived at this in very different ways. So Gerby and Philippe J's lab in France, they really had been looking at the sort of cell biology and differentiation of tuft cells. Michael Howard and Wendy Garrett's lab, they came at it from more of a microbiology angle. And all of us really, I think, came to the same conclusions about this Tuft ILC2 circuit, which really defined Tuft cells as what we kind of think of as a missing link in this type 2 immune response that are ideally positioned here to sense the lumen and communicate to the cells down below. So one of the really important contributions of Michael Howitt's paper was to add another microbe to the list of microbes that activate this circuit, specifically these protists called tritrichomonads. And I want to just take a minute to sort of highlight this paper because it'll be important later on. This was one of these stories where they realized that the mice they were working with in their facility at Brigham's were not like the ones they were ordering from Jack's. And they traced this back to the observation that their in-house mice were abundantly colonized with these protists, tritrichomonad protists, so named because they have 3 flagella. And they did a really nice experiment where they were able to add just these protists to the Jack's mice and show that that was sufficient to induce the same hyperplasia that we were seeing with the helminth infection. So an increased number of Tuft cells in the small intestine. Okay, so that was sort of the state of our knowledge when I started my lab. And what I want to talk about today are 2 stories related to what we've been working on. One is really a detection story because the circuit is all fair and well, but you still need a way to tell the immune system that these agonists are there. So I'll talk about that. And then also we'll talk about what else Tuft cells do besides just making IL-25. All right, so let me start with the sensing side of things. And the evidence that Tuft cells were actually directly chemosensory emerged at this point more than 20 years ago when people who were working on chemosensation, taste transduction, were developing tools for their research and realized that they were able to identify cells in other tissues that were also labeled with these markers. So for example, this antibody for GNAT3 was also lighting up Tuft cells in the intestine. This reporter mouse for TRPM5 was lighting up Tuft cells in the intestine as well. And so what eventually became clear is that this entire chemosensing pathway that had been really worked out in the context of taste transduction was present in these Tuft cells, also called brush cells or solitary chemosensory cells in other places that they expressed the signaling pathway. So when I talk to immunologists, I need to spend lots of time explaining this. You are all, of course, really familiar with it. But just to re-emphasize that you have on the cell surface your taste receptor, a GPCR, often coupled to the specialized G-alpha, GNAT3, signaling through PLC beta 2, releases calcium, and activates the calcium-gated TRPM5 channel that is a cation channel and allows for depolarization of the cells, release of things like ATP and acetylcholine to drive taste transduction. So again, it's really these core components of this chemosensing pathway that are expressed in Tuft cells, and Michael Howitt again reported this first, that in a TRPM5 knockout, when they colonize with these tritrichomonas protists, you do not get the Tuft cell hyperplasia. So that really implicates chemosensing in the detection of protists. And we have now done this also with the worms, the Nipostrongylis, the TRPM5 knockouts. Here's the normal Tuft cell response that you see. It's severely attenuated without TRPM5, and those TRPM5 mice fail to clear the worms in a timely manner all the way out to day 14 here. Okay, so clearly an implication of chemosensing, but still a question about receptors and ligands. And so Maria Naj-Sabati and Jack McGinty were the first graduate students to join my lab. And actually during their rotations, we sort of made the first observations with Succinate. And so I asked them to work together to try to take that on in their first year. And they did a really amazing job of as young graduate students turning this around and getting this completed really quickly. So the question that we were asking and they were asking is, what are the receptors upstream of TRPM5 that activate this circuit? And again, we had some data that I generated when I was still a postdoc with Richard Loxley with help from other postdocs, Christoph Schneider, Corey Miller, Josh Pollock, where we sorted, using our reporter, sorted Tuft cells from different tissues, thymus, trachea, gallbladder, small intestine, colon. We also included non-Tuft epithelium from the small intestine as a reference, and used this then to generate a a core signature. There were, of course, tissue-specific differences, but we wanted to know what was common to Tuft cells in all locations where we could sort them out. So we had defined this core signature of about 100 genes, and IL-25, of course, is there. That's what we use to sort them. But notably also, again, all of those core intracellular components of the chemosensing pathway, TRPM5, GNAT3, PLC beta 2, for example. And so the question then was, well, what's the receptor? And the sort of simple and a very attractive idea was that Tuft cells are just tasting worms in the intestine, and they're using taste receptors to do this. But when we actually looked at the expression of the canonical taste receptors in the different tissues, we could see some of them, consistent with previous publications too. We could see some of them expressed in the thymus and the trachea. But we see absolutely no expression of either the TAS1 or TAS2 family GPCRs in the small intestine. And so we started to think about alternatives, and given that the core pathway is there, perhaps you can plug a different GPCR into that. That was our thinking. So we looked at other GPCRs that were expressed at high levels in Tuft cells in the intestine, not so much in their neighboring cells. And we picked out two that were linked to metabolite sensing that were interesting to us. One is FFAR3, a receptor for short chain fatty acids, really highly expressed in Tuft cells in the small intestine, much lower in the neighboring cells. And the same was true for succinate receptor, which is the surface receptor for the citric acid cycle intermediate succinate. And so we did the very simple experiment. This is the one we did during Jack and Maria's rotations of just putting these ligands for the receptors in the drinking water of the mice and looking to see what happens. And I didn't think this would work, to be honest, but it turns out that succinate in the water was sufficient to induce a Tuft cell hyperplasia. You can see it here and quantified here. Now we're giving very high concentrations for 7 days. We don't know what the concentration is effectively in the intestine, but just to be clear, these are high concentrations. And we don't see anything when we use the FFAR3 ligand. So that's a receptor we, at this point, don't have any understanding of what it's doing in Tuft cells. But the succinate result was, of course, very exciting, and we wanted to pursue it. One of the first things we were concerned about or wanted to evaluate was whether this was actually a direct effect or whether the succinate was altering the microbiome, and then that alteration is what was being detected. But when we repeated the experiment with succinate in the drinking water in germ-free mice, we got exactly the same result. So this really suggested a direct detection of succinate by the host. And it wasn't just this Tuft cell response that we could replicate with succinate. We got cell hypertrophy, so that you can see them here full of mucus, so their size is bigger. Again, a hallmark of a type 2 immune response. We could also look in the draining lymph nodes of the small intestine and find accumulations of ILC2s, eosinophilia, again, hallmarks of type 2. And if we pre-treated mice with succinate and then infected them with the worms, we didn't prevent colonization, but we did speed clearance. So you start to see some clearance by day 4 or 6, which is much earlier than in unmanipulated mice. So all in all, it seemed like the single molecule succinate was sufficient to induce a bona fide type 2 immune response in the small intestine. And so then we, again, wanted to sort of pick apart the components of this circuit drawn as a cartoon here. And I'll just walk through these quickly, but we had a lot of knockout mice available. So if you delete IL-13 signaling, therefore you can't get this expansion of Tuft cells, so nothing happens. Without ILC2s in these mice, nothing happens. RAG for the immune aficionados, this deletes T cells and B cells. They're partially required. IL-25, again, completely required. ST2, this is the receptor for IL-33, another signal that activates ILC2 is not required. TRPM5, which of course is this chemosensing channel in the Tuft cells, is required. And then finally, we generated succinate receptor knockouts, and that was again required. So this response is being mediated through the circuit. It's not, we did other experiments to show that it's not succinate signaling directly on the stem cells, and it's not succinate directly inducing replication of the Tuft cells. It's this activation of this feed-forward circuit that drives the response. And lastly, in the POU2F3 knockouts, of course, we can't count Tuft cells, but we can count goblet cells. And again, we see a failure to activate the response. Okay, so that was all fair and good, but what really is the physiologic source of succinate? This was a completely non-physiologic experiment to this point. And so we started to think about the anaerobic environment of the small intestine and the metabolism that occurs there. In mammals, of course, under anaerobic conditions, we produce lactic acid. Yeast famously makes ethanol and CO2. But bacteria have a much more diverse set of approaches to handling this anaerobic environment. And they will produce a wide variety of metabolites. And among them, you do find bacteria that will secrete succinate as a waste product. And we went into the literature, found a really nice review from Miller and Martin with some evidence that helminths and tritrichomonads might also secrete succinate, but we wanted to test this ourselves. So we put either Nipistrongus braziliensis or tritrichomonads into a dish and waited overnight and measured the metabolites that, that were produced. And succinate is not the dominant metabolite of either of these, but it is definitely present. So we could easily detect succinate made by both the worms and the protists. And so then we went back in vivo and we started by testing a requirement for the succinate receptor in a worm infection. And we were excited at the time. We thought we had sort of solved this longstanding mystery about detection of worms, but the clearance of the worms and the succinate receptor knockout really was essentially normal. The Tuft cell hyperplasia was completely normal. And really we've to date found no in vivo requirement for succinate sensing in a helminth infection. That was surprising, but we maybe have a better understanding of that now. But when we tried the same thing with the protists, now we put the protists in, normally you get this response here. Without the ability to sense succinate, that's completely gone. So is the expansion of the ILC2s. So the detection of the protists requires a succinate receptor, but the detection of the helminths does not. What about bacteria? So I mentioned just a minute ago that bacteria are known to produce and secrete succinate. The thing is that in a sort of healthy, balanced microbiome, we wouldn't expect there to be a lot of free succinate around because of this process of syntrophy or cross-feeding. So you'll have primary fermenters like Bacteroides that will use carbohydrates and can produce succinate. But then secondary fermenters like Clostridia often will use the succinate and secrete short-chain fatty acids. But instead, you could imagine scenarios in which the balance between these is disrupted, and now you can accumulate succinate. And really, it was Pei Hua and his lab who started thinking about this first and published this in their really nice PNAS paper, where they induced a dysbiosis either using streptomycin or by pegylating the water, because the Sonnenberg lab had previously shown that both of these treatments will increase cecal levels of succinate. And indeed, they found a Tuft cell hyperplasia that was completely dependent on the receptor. But they noted, and this will become important again in a second, that this really was restricted to the very terminal ileal segments of the small intestine. So we've sort of explored this bacteria function a little bit further. We haven't done a lot. We started out by trying to do the same experiment with the streptomycin, and in our colony, it doesn't work. So I think this is really dependent on the microbiome. You probably need some strep-resistant bug that makes succinate that you can expand or something like that. I don't know. I certainly believe that this happens, but in our colony, it didn't. So what we did instead was a really reductionist experiment where we took a known succinate producer, Bacteroides ovatus, and we were really lucky to get from our collaborator, Megan Baldridge at Washington University, a mutant. These are putative succinate transporters, and these mutant ovatus have a 50% defect in succinate secretion. And so we took these two strains, gavaged them into germ-free mice, and waited 7 days and then counted Tuft cells. We confirmed that both of these, both the wild type and the mutant, colonize at equivalent levels, but only the wild type really induces a Tuft cell hyperplasia response, like you see here. And what's interesting is just like Pei Hua's lab found, the closer you go to the cecum, the more pronounced this becomes. So if you count just the last 2 centimeters, you see a much more robust hyperplasia occurring there and nothing with the mutant. All of this being consistent with where bacteria predominate in the small intestine. And what we've really sort of come to appreciate is that there's a regional specificity to the succinate receptor, the succinate sensing. So if you think about our initial experiment with the drinking water, we're giving the drinking water, through the stomach, and the concentration of succinate will actually be highest right by the stomach. And despite that, we see a much bigger response in the distal small intestine than in the proximal. And we think this is predominantly because the expression of the receptor is much higher. When we sort Tuft cells from the distal small intestine, it's about 10-fold or more higher than in the proximal small intestine. It's not absent in the proximal small intestine, but it's this sensing predominates here in the distal small intestine, and is also where the bacteria and the protists reside. The worms, on the other hand, reside predominantly up here by the stomach, and this may help explain why in vivo we don't see any link with succinate sensing. Okay, so just to summarize then this first part, and again, this was work that we published at the same time as others, Peiwa's paper and also a paper from the Loxley lab. But again, we all basically reported the same thing. Succinate is a molecule that is sufficient to activate Tuft cells and the circuit through the succinate receptor. We think there's a calcium flux that opens TRPM5, and you get the downstream activation of the circuit. We have some pretty clear evidence that this is required for sensing of tritrichomonads. Less clear when and in which context the bacteria might be doing this, but it's certainly possible, at least in that reductionist experiment we did in the germ-free mice. And intriguingly, although TRPM5 is absolutely required for detecting helminths, there's no requirement for succinate. It could be redundant. We've tried some double knockouts and never found any phenotypes. At this point, we suspect there's just another receptor that we have yet to identify. Okay, so that is the first story about this upstream chemosensing. And so let me switch now to talking about what happens downstream of Tuft cell activation. And just intuitively, it seems very unlikely that the only reason these cells are here is to secrete IL-25. They must be doing other things. But we have actually experimental data to support that idea. So we compared here wild-type mice to IL-25 knockout mice to POU2F3 knockout mice, again infecting with Nipostrongylus and tracking worm clearance. So wild-type mice have all cleared by day 10, but the two knockouts are both still colonized. But if you go out further, you start to see them diverge. The IL-25 knockouts have essentially cleared by day 14, and the mice that entirely lack Tuft cells are still colonized even at day 21. So direct evidence that Tuft cells do more than just make IL-25 in terms of their contribution to the type 2 immune response. So what else could they be doing? And now I need to take a quick sort of side trip back into ILC2 biology, and I'll try to explain this as clearly as I can, because I know this is not really the expertise for all of you. But we started to make sense of all of these various receptors that contribute to ILC2 activation by thinking analogously to T cells, where of course people have been studying T cells for much longer than ILC2s. And we know that in T cells, you have a requirement for sort of survival signals, IL-2 and IL-7 that mobilize STAT5. And then you have a T cell receptor. That's what defines a T cell. And the signaling that we teach everybody in introductory immunology classes is that this receptor alone mobilizes 3 critical transcription factors: NF-kappa B, AP1, and NFAT. Those all go to the nucleus, and they cooperate there on the promoters of the various effector molecules to drive expression. So the thing that defines an ILC2 is that they don't have a T cell receptor. In other ways, they're actually very similar to Th2 cells. They make largely the same effector cytokines. They have the same master transcription factors regulating them. And so we were wondering, how does an ILC2 replicate this T cell receptor signaling? And it became clear to us that you could make sense of these receptors by grouping them based on the transcription factors that they predominantly activate. And so the optimal way to activate ILC2 is to kind of get one thing from the STAT5 family, add the STAT6 signal, get an NF-kappa B and an NFAT. And we sort of showed this in other work that I did as a postdoc, where we were combining IL-33, an NF-kappa B inducing signal, with leukotrienes, LTC4 and D4, that mobilize NFAT. And we showed that when you combine these two together, you get this really massive synergy because they have non-redundant contributions to activations of ILC2s. Okay, so why am I telling you all this? Well, because I want to focus on leukotrienes now, and these are lipid-derived inflammatory signaling molecules. They are synthesized very rapidly, within minutes, on demand. The synthesis is initiated by activating a phospholipase A2. Often, this is a calcium-dependent enzyme, but there are many different ones. This liberates arachidonic acid from membrane phospholipids, and this is then a substrate either for the cyclooxygenase enzymes that make prostaglandins, or for 5-lipoxygenase or ALOX5, which makes leukotriene A4, and that is then further converted into the chemoattractant LTB4, or into this family of cysteinyl leukotrienes, C4, D4, and E4. And the receptors that they signal through are shown down here. So really one of the important things to understand here is that they're different than proteins because they are made rapidly and they're also degraded rapidly. So these are very labile signals for immune responses. Mostly they historically have been shown to be made by myeloid cells, mast cells also, and they have very pleiotropic effects. So all of these have receptors on lots of different cells. The cysteinyl leukotrienes specifically have receptors on smooth muscle, on the endothelium, on hematopoietic cells, and of course on ILC2s. And the thing that was immediately apparent to us and many other people is that part of the core signature of Tuft cells includes all the enzymes to make leukotrienes. So you have ALOX5 here, and an accessory protein for ALOX5, an LTC4 synthase, which makes the cysteinyl leukotrienes. All Tuft cells and all tissues express these. And so the very obvious question, sort of combining what we knew about ILC2s and what we knew about Tuft cells, was to ask whether Tuft cell-derived cysteinyl leukotrienes contribute to ILC2 activation. And this is work done now by Jack McGinty. You saw him before. He contributed to the Succinate work, but this was really his story that he was really the driver of, and he deserves an enormous credit for the work that he did here. So before I show you the data, just again introducing a couple of tools. One is what's called the SMART13 reporter. This again was originally generated in the Loxley lab. Every cell that is making IL-13 will put a mutant human CD4 protein on its surface. And it's there just as a marker that we can then stain by flow cytometry to quantify the number of cells that are making IL-13 and roughly how much they're making by measuring the MFI. And so you've already heard about Nipostrongylis. I guess the key thing here is that the larvae enter through the skin, traffic through the lungs and the intestine, but it's an acute infection and it's cleared within 8 days. We now add another model, another Helminth model, the Lignosomoides polygyrus. Here the infection is directly fecal oral, so you just gavage the infectious larvae into the mice. And this is a chronic infection in mice, so it establishes long-term infection. So they're nice to kind of compare the two. Okay, so the first thing Jack did was just to confirm in vitro that we had worked out the activation of ILC2s by leukotrienes in the lung previously, but the ILC2s can differ a lot between different tissues, and we wanted to make sure that everything worked the way we thought in the intestine. So he sorted the ILC2s out of the small intestine, stimulated them with various leukotrienes, and just as in the airways, LTC4 and D4, these two here that signal through their cysteinyl leukotriene receptors, potently activate IL-13 production in ILC2s. E4 and B4 don't really do much, which is not unexpected. And again, just as in the airway where we were combining 33 and the leukotrienes, here in the intestine, when we combine 25 and the leukotrienes, we get this really nice additive or even perhaps synergistic effect of the two signals together. Again, because the leukotrienes can mobilize NFAT in ways that IL-25 cannot. All right, so then switching in vivo, Jack started out using ALOX5 whole body knockouts. And again, this is the upstream enzyme here required for production of all leukotrienes. And we wanted to make sure that nothing was wrong with these mice homeostatically. The number of ILC2s in the small intestine was completely normal. We've also sequenced them transcriptionally. They look just like wild type. The small amount of IL-13 that they make constitutively was normal without leukotrienes. So these mice seem to be normal homeostatically. But then if you start to activate the system, you see the defects. And here Jack invented a really nice assay that I haven't seen anyone else do before, where we wanted to ask about the earliest initiation of type 2 immunity. And Nipostrongylus is a bad model for that because the infection is actually in the skin and we have no idea when the worms really get to the intestine. So he used Haligmosamoides instead, where he could gavage it in and just wait overnight and then take the ILC2s out the next morning and use the IL-13 reporter to look at their activation. And you can see here's the control. They make very little without any infection, but just hours after putting these worms in, you can start to see IL-13 production by the ILC2s. And that IL-13 production is dependent on TRPM5, so chemosensing by Tuft cells. This is a whole body knockout, but the only cells in the small intestine that express TRPM5 are Tuft cells. Also dependent on IL-25, and largely dependent on ALOX5, so on the production of leukotrienes. Again, no requirement for IL-33 signaling here. But you can see the quantification on the right. All right, so that was just overnight. But if Jack then extended this out longer and waited 4 days, so now that initial activation of the ILC2s has produced IL-13, and the IL-13 is signaled in the stem cells and changed the output of the stem cell compartment. So by day 4, you start to see, especially down emerging from the crypts here, this accumulation of Tuft cells. But that is completely absent in the absence of leukotrienes. And even 14 days post-infection, there's still a defect in the Tuft cell number in the epithelium. And it's not just Haligmosamoides. We can do this with Nipostrongylus as well. We're looking here either in the proximal small intestine where colonization first happens, 5 days post-infection, and the hyperplasia that you normally see is essentially absent without leukotrienes. And distally, at the end of the small intestine, also a defect at 7 days post-infection. And just as before, this is not just a change in the Tuft cell numbers, it's really the entire type 2 immune response in the small intestine that's defective. You don't see this expansion in the size and the number of goblet cells if you don't have the leukotrienes. You don't get the activation of the ILC2s in the lymph nodes. And all of this translates into a defect in worm clearance. So here, especially on day 7, where most of are clearing, the ALOX5 knockouts are all still fully colonized. Now, I will note that by day 14, they've cleared, and this is really, we think of this as a kinetic defect. There's a lot of redundancies in this system, and certainly other things can take over if you don't have the leukotrienes. But in the early rapid response, there seems to be an important role for them. So let me just address a few caveats at this point. So again, we were using a whole body knockout initially, and so that doesn't tell us which cells are making the leukotrienes, and I'll address that in just a minute. But the other issue is that it doesn't tell us which of the leukotrienes are really the relevant ones. We've eliminated all of them. And so Jack did a couple of experiments. One was to use this drug, montelukast, that is a specific inhibitor of cysteinoleukotriene receptor 1. And we knew from previous in vitro experiments that this is the dominant receptor for C4 and D4 in ILC2s. And doing that, he could replicate this phenotype at 16 hours post-infection with Haligmosamoides, where you have a defect in the activation of ILC2s without cysteinoleukotriene receptor 1 signaling. And we also were able to obtain knockouts for that receptor. And again, those completely phenocopy the entire deletion of leukotrienes. So this doesn't rule out a non-redundant role for other leukotrienes, but it certainly implicates a critical role for C4 and D4 signaling through cysteinoleukotriene receptor 1. The other caveat has to do with this phenomenon called shunting, where people have shown that in these biosynthetic pathways for lipids, when you eliminate an enzyme, you can accumulate upstream products that then are shunted into other pathways, in this case, the shunting would predominantly lead to increased production of prostaglandins. And so Jack used ibuprofen to inhibit the cyclooxygenases here and found that that made no difference. So the phenotype that we were seeing in the global knockout didn't seem to result from some accumulation of prostaglandins. Right. So, but then let's address maybe the most important question: what are the physiologically relevant source of the leukotrienes, and is it Tuft cells? So, excuse me. I mentioned that as part of the transcriptional signature of Tuft cells, they have all the enzymes to make leukotrienes, but we wanted to confirm this at the protein level, so we did that with an antibody for ALOX5, and here's a sort of a zoomed out view of the small intestine with, you can see the villi, and the red cells here are all Tuft cells, and you can see that all of them essentially have some ALOX5 staining. This one not, but I think that's probably just the way that the section was cut. There's definitely also ALOX5 in the lamina propria and these immune cells, myeloid cells down here. Those are the kind of the canonical source of these leukotrienes. But in the epithelium, the only cells that we see that are ALOX5 positive are Tuft cells. And if we zoom in, we see an interesting relocalization. If you compare uninfected to infected, it goes from a largely nuclear, there's some cytosolic that's masked by the signal here, but during infection, this all relocalizes to this perinuclear ring. And that is well defined already in the ALOX5 literature as the site of leukotriene synthesis. So that's where this enzyme goes when you are making leukotriene. So this is consistent with an activation of the pathway. And also we were able to use this antibody to together with another Tuft cell marker, phospho-EGFR, using human biopsies, largely healthy tissue discarded during Whipple surgeries and the like. And we can find Tuft cells, and they're all ALOX5 positive in the human duodenum as well. So that confirms that the cells have all the things they need to make leukotrienes, but we also wanted to actually show directly production of it. And in vitro studies of Tuft cells are difficult, as many of you know, especially when you take them from the intestine, because the intestinal epithelium just doesn't like to be cultured in vitro. So lots of people have been making these three-dimensional organoids, but those present a lot of challenges, especially if you want to stimulate them apically. That side is encased inside the organoid. So what we did instead is we coated dishes with Matrigel, put small intestinal crypts on them, and they lay down flat and form monolayers that include all of the intestinal epithelial cells. They're not replicating, they're not super happy, but they're happy enough that we can incubate them overnight and then stimulate them acutely the next day. And so what Jack did initially was just use a calcium ionophore ionomycin to non-specifically activate leukotriene production. And he can see really nice, by ELISA, nice production of cysteinoleukotrienes in the wild-type monolayers. But using monolayers from POU2F3 knockouts, which have no Tuft cells, but still have all the other, they have goblet cells and enteroendocrine cells, completely loses the signal, the leukotriene signal. So there's clearly a Tuft cell dependent production of leukotrienes. It also requires ALOX5 as expected. And kind of an intermediate phenotype for TRPM5 that we don't understand at this point. We don't even know whether this is a defect in production or secretion of the leukotrienes. We hope to figure that out. And then more physiologically relevant, Jack was able to take worms, collect all of the secretory product that they release, the so-called NES, and that was sufficient, again, to drive leukotriene production in these monolayers, but not if you lacked Tuft cells. So that really suggested a chemosensing of a worm product that was activating Tuft cells, leading to production of leukotrienes. And then lastly, doing the in vivo experiment, we generated ALOX5 floxed mice, crossed them to various Cres. We started out with a villain Cre to delete ALOX5 from all of the intestinal epithelial cells. But again, only the Tuft cells express ALOX5. And remarkably, this really completely phenocopied the global knockout of ALOX5 in terms of the Tuft cell numbers. And then we were really lucky that Ichiro was, again, incredibly generous to share the POU2F3 Cre ERT2 mice that he had made, so that we could specifically delete ALOX5 just from Tuft cells acutely, so with administration of tamoxifen. And again, this phenocopies the global knockout. As some important controls, we also tried deleting ALX5 from mast cells with CPA3-CRE or from myeloid cells with lysem. These are, again, canonical sources of leukotrienes, but at least in this context, even though there are certainly lots of ALX5 producing cells in the lamina propria here, they don't contribute to this response at all. And it's not just the Tuft cells. We get the same result if we look at the goblet cells. Either the Villain-CRE or the POU2F3-CRE phenocopies the global knockout. And worm clearance, same thing. A delay in worm clearance in the global knockout also when we delete ALX5 just from Tuft cells. All right, so I'm gonna add one last twist now, although I think this is actually I find this really interesting, but it also is a bit of a head scratcher for us. So you may have noticed that everything I've told you about leukotrienes was in the context of helminth infection. What about succinate and protists? And what's been really interesting but surprising to us is that in this context, we find no requirement at all for leukotrienes. So no matter how we look at this, if we use that rapid assay where we just give succinate acutely for 36 hours and look at the ILC2 activation, that's completely normal without leukotrienes. If we give succinate for 7 days and delete the leukotriene production just from Tuft cells, completely normal. And if we actually use the protists, we transfer them into these mice, we get a hyperplasia, but it's completely normal without leukotriene production. And so there's, just as we saw a split in terms of the succinate sensing between the protists and the worms, we're also seeing this dichotomy with the role for leukotrienes in the two responses. And we've started to try to understand why this is. One possibility we considered was that there's another molecule that's been shown to be really important for ILC2 activation in the intestine called NMU, a neuropeptide. And it, just like leukotrienes, uniquely can activate NFAT. So we thought maybe it was responsible, it was sort of the signal too for succinate, but when we got the knockouts and treated them with succinate, everything was completely normal. So redundancy is still possible. We're making the double knockout, but at this point, no role. The other thing we considered was maybe just like the succinate receptors is expressed at much higher levels distally than proximally, maybe this similar thing going on with the leukotriene production, but no, we see all of the Tuft cells, again, express all the enzymes, but despite that, we don't see a requirement in the succinate pathway. All right, so let me end with this summary here. I think this graphical abstract sort of has everything I told you. The Tuft cells are really sitting here as immune sentinels at the barrier. They monitor the lumen, just like really had been shown nicely in the airways earlier, and they communicate down to the underlying tissue. All of this requires TRPM5 and IL-25, but in the case of the helminth infection, there's a receptor and a ligand that we don't yet understand. In the case of tritrichomonads and perhaps bacterial dysbiosis, this is mediated by succinate. And then despite the fact that all these cells have all the machinery to make leukotrienes, only when you detect the worms is there a requirement for this. And you know, I'd be really interested to hear from all of you who have way more insight into the kind of the chemosensing pathway and the intracellular things here, if you have thoughts about how it is that activating these two different GPCRs could lead to potentially to different outputs. We don't know 100% for sure that succinate activation of the Tuft cells does not lead to leukotriene production, but in our hands, it looks like it doesn't. And then just to kind of highlight emerging principles here. So, you know, so specifically in the distal small intestine, the Tuft cells are monitoring this microbial succinate level, but we don't even really understand why. We thought this would have something to do with helminths, but it doesn't. And so we think that maybe this is some sort of pattern of parasitism or dysbiosis. This increased succinate level tells the immune system that the microbial metabolism is out of balance. And for some reason, it then decides that a type 2 immune response is appropriate, but we don't really know why. And the other thing is really about the expanding understanding of diversity of these Tuft cells. So, you know, clearly the receptors they use just based on transcriptional analysis differ between different tissues. But now we're finding that even the effector functions are different, even within one tissue, that you have this, and perhaps this has something to do with tuning their output to the kind of the level of danger that each of these represents. So the helminths maybe are more of a danger and therefore require this additional leukotriene signal, whereas tritrichomonads are really commensals, and the idea is to just kind of live with them rather than to try to expel them. But those are all speculations at this point, but happy to hear other thoughts, or hopefully I'll just spur some discussion. And lastly, we've all been trying to find new ways to interact with our labs. Sometimes they decide to sit out some of our Zoom meetings, but they're actually a great group of scientists, and I've been really lucky to work with them and to have them join me. I mentioned Jack McGinty and Maria, the first graduate students that drove all of the work that I showed you, but we have other people doing other exciting things. Great collaborations with my old lab, of course Ichiro with the mice, and others providing reagents and funding that's been really critical. So I'll stop. I can leave this up or I can stop sharing. I don't know what you prefer. You can let me know. But thanks for listening.
Thank you, Jakob. Great talk. Questions from participant audience? So there's a question. I don't know, Jakob, you saw it in the Zoom group chat.
Oh, here we go. Okay, Andy Vaughn asks, have you seen evidence of tuft cell expansion in non-Th2 types of pathogens? Yeah, I guess the first thing I'll say about that is we have never seen tuft cell expansion anywhere other than the small intestine. I guess you could, actually, I mean, Andy's discovery of the tuft cells in the distal airway after flu maybe is one example. I don't know if it quite counts as expansion. So this real hyperplasia that we see, we only see that in the small intestine. We never see that in the colon or the cecum. And we actually haven't spent as much time studying those. In the small intestine, yeah, I mean, only with some bacteria and some dysbiosis, you see some tuft cell expansion. We haven't spent a lot of time looking at anything like, say, salmonella or other bacterial pathogens. There's not a lot of great models for small intestinal bacterial infection in mice. Most of it's in the colon. But yeah, it's a good question. I mean, you know, this is clearly not just a type 2. I mean, it's a type 2 immune response in the sense that those are the molecules that are activated, but it's being used in what seem like not classically type 2 settings, especially in the distal small intestine with the protists. And at this point, we don't really understand what that's about. Okay. Question, are intestinal tuft cells cholinergic and do they release acetylcholine in response to activation? Yeah. Yes, so as many of you know, tuft cells, brush cells, solitary chemosensory cells in other organs are chat positive and cholinergic. In the intestine, chat is also part of that core signature that defines all the tuft cells we've looked at. And we've used the reporter, and most of the tuft cells, although not 100% of them, are reporter positive in the small intestine. We don't know the function of that acetylcholine. In the airways, I think there's good evidence that at least in some parts of the airways, there is direct crosstalk to neurons, work done by Marco Tizzano, for example. Based on just on imaging, we have had a really hard time seeing any clear evidence of sort of co-localization of neurons with tuft cells in the small intestine. And so we're thinking about other possible targets of that acetylcholine and other functions, but I don't know at this point. Another question, is there any evidence that pH is a factor in activation of tuft cells? Yeah, I love that idea. And we spent a little bit of time exploring that early on, thinking along the lines that maybe, you know, if you have a worm infection, you change the pH of the intestine and that's what's being detected. At this point, we have no evidence for it. And there's, yeah, I guess that's all I can say. We don't have any evidence for it. Do viruses interact with Tuft cells? Yes. So really the best example for that is norovirus. There was really nice work done by Craig Weiland in Skip Virgin's lab, showing that certain strains of mouse norovirus replicate exclusively in Tuft cells. It's actually quite remarkable. So mice without Tuft cells cannot be infected with that strain of norovirus. So they express a lot of the receptor, so that explains how it gets in there, but why that is a niche for the norovirus, I don't think it's clear. I don't know of any other evidence of direct infection by other enteric viruses. Also, it's not clear, well, yeah, no other examples that I can think of direct sensing of viruses. Would you expect Tuft cells and mucin cell expansion if you cause inflammation or changes of the microbiome somehow? Yeah, so I think in the right circumstances, yes. I think that Peiwa's data with the streptomycin and the pegylation where you induce a dysbiosis and then you see it, I think that really supports this idea that when you have increased levels of succinate because the microbiome is out of whack, that's when you could activate this circuit and see all those things. Now, there could also be sensing of things other than succinate that the microbiome produces, and that could certainly be occurring even in the context of helminth infection, but we don't know. Let's see, 2 natural experiment questions. 1, people take anti-leukotriene medicines for asthma. Would you expect them to have altered gut immunity? Yeah, exactly. We've speculated about that. There's a montelukast inhibitor that we used in mice and that is often used in the clinic. Yes, we would expect that if they were infected with a helminth, it might be altered. Of course, that at least, you know, in our communities is not really often an issue. And if they had some reduction in allergy, that probably wouldn't show up in intestinal allergy. So, but yeah, certainly could be involved. And then number 2, some fermented foods like sauerkraut and some cheeses are high in succinic acid. With these enhanced gut immune function and altered gut microbiomes via Tuft cells. Yeah, also presumably, I mean, succinate is in all of our cells. It's in all the mitochondria. So I guess you could argue that maybe if you eat meat, that has a lot of succinate in it. We don't think that dietary succinate is going to activate this. Again, we were giving 150 millimolar in the drinking water, so extremely high levels. So I think any dietary direct consumption of succinate is unlikely to activate this. Now, your diet, of course, can change your microbiome, and it may alter the succinate production by the microbiome, and that could certainly be detected. And then, I'm wondering where the short-chain fatty acids, succinate, propionate, butyrate, acetate, measured with time, were they measured with time in the drinking water? Might the other short-chain fatty acids not have induced Tuft cell hyperplasia as succinate because they may be more volatile and were less were acutely ingested by the mice? Yeah, so that's a good technical consideration. We sort of took our cues from earlier work done with short-chain fatty acids in drinking water by Wendy Garrett's lab, and they actually made some efforts to measure the stability over time and found that within a week, there wasn't a whole lot of loss. So at this point, we think we're probably still have enough in there, but it's, we really, yeah, we tried to measure them in the intestine and couldn't really determine the differences. So hard to say what's actually getting in there. And it's always hard to interpret a negative result. So there could still be a role that we've missed at this point. Yeah. Okay.
So Jakob, I have a question for you. So
Yeah.
we know like NB is a pretty accurate, generate accurate response. At the H. pylori gyrus seems like a chronic. Why?
Well, I mean, I guess, so the Nipostrongylus is a rat-adapted parasite, so I guess you could argue that it hasn't learned to evade the mouse immune response as effectively, although it's hard to see what's so different about them. I think really the main thing is that for Lignosomoides, it has all kinds of immune modulatory functions that have been really beautifully studied by other people, mostly in England, like Rick Maisels. One of the things they make is a TGF-beta mimetic, so they induce Tregs, and they dampen the immune response in that way. They also inhibit IL-33, this molecule that's really important for the type 2 immune response. So in that case, they seem to establish the chronic infection because they are immunomodulatory. And accordingly, the Tuft cell response is less. It never gets to the same threshold as you see with Nipostrongylus. Yeah.
So, but the primary infection seems like chronic, but if you do the secondary infection, seems like acute. Oh, what do you think of that with the H. pylori gyrus?
The secondary, yeah, so then it is cleared acutely. There is a memory response, and that seems to be, I think the difference there is that it's more vulnerable during the colonization phase. So it has its life cycle includes this granuloma phase in the tissue where it has to develop into an adult. And so I think in a primary infection, the immune system just doesn't get going fast enough to block it before it becomes an adult. But in a secondary, the response is so fast that I think it can act on that or on those early stages. Yeah. I guess there's one more question here. You started out talking about Tuft cells expressing FFAR3. Any thoughts about fatty acid production by microbes acting on Tuft cells via FFAR3? Yeah. I mean, all I can say is, you know, putting in the drinking water didn't do anything. We have FFAR3 knockouts. No phenotypes in any of our models. We've made FFAR3 succinate receptor double knockouts and still no phenotype with helminths. You know, it's always hard to know what G alpha proteins these GPCRs are actually coupling with in any given cell, but from the literature, it seems like FFAR3 may couple more readily with G alpha I's, and so maybe it has more inhibitory function. Although, again, it's not like in the knockouts we see an enhanced response. So we've been really stumped on that one. Enteroendocrine cells also express FFAR3, so it's not just Tuft cells. But if anyone has thoughts about an experiment, we have the knockouts.
So any other question, comments? If not, let's thank Jakob for this great talk. Thank you, Jakob.
Yeah, thank you. And Phil, you know, anyone feel free to email me or we can set up another time to talk. But I really appreciate the invitation and hope we all get to meet in person someday soon.